The overall goal of this procedure is to observe the dynamic interaction between a single DNA binding protein and a DNA molecule. This is achieved by combining, in a custom-built microscope, two single-molecule techniques: double optical tweezers and fluorescence microscopy. This setup is also equipped with bright field illumination for visualization and nanometer stabilization of the sample. As a second step, a multi-channel flow cell, integrated in a microflow system, is built to allow up to 4 laminar flows to efficiently recruit and assemble all the components required for the experiment. Next, the single-molecule construct is assembled in order to perform the experiments. First, 2 streptavidin-coated microspheres are caught with the dual optical tweezers in the first channel. The 2 trapped beads are then moved to the second channel to allow the attachment of the DNA template, labeled with biotin on both extremities. The presence of a single DNA molecule is checked in the third channel by a DNA force extension analysis. Lastly, the bead-DNA bead construct, named DNA dumbbell, is moved to the protein channel, where the fluorescently labeled lactose repressor protein is present. Results are obtained that show diffusion and specific binding of single LAC-I molecules on a single stretched DNA molecule, and the quantification of the kinetic parameters of LAC-I motion.
The main advantages of this technique over existing single-molecule methods are the possibility to control tension applied to the